My name is Akaash Kumar, and I'm a final year PhD student at the MRC Laboratory of Molecular Biology and the University of Cambridge. And today I'm going to give you all an overview into my PhD project on advancing imaging technologies to visualize more in living systems. So firstly, I would like to say that it's an absolute pleasure to be here presenting the technology that I've developed during my PhD. And this enables, for the first time, simultaneous visualization of many biological features within living systems such as cells. But before I introduce my technology, I just want to give you a bit of background to something called fluorescence microscopy. Now, fluorescence microscopy is a fundamental technique that is used to study a wide variety of biological systems by a wide variety of industries. And these industries range from academic research through to pharmaceutical development and medical diagnostics. And thus, it is an incredibly important tool to study science. And the global fluorescence microscopy market was valued at $500 million in 2019. Now, biological systems such as cells are highly crowded environments with millions of proteins and structures. Therefore, to study a biological question, one must be able to see the biological target of interest, which can be a bit like finding a needle in a haystack. Now, the overarching idea of microscopy is to produce an enlarged image of the sample, in this case, 2 cells. But we want to specifically see particular structures within the very crowded cell without seeing everything else, which is like seeing a needle without seeing any of the hay. Now, through genetic engineering or chemical modifications, scientists can attach a fluorescent tag to a biological structure of interest within the cell. We can then hit this fluorescent tag with a certain color of light, and they then emit light in a specific color range. And this enables the visualization of the biological structure of interest in the sample without seeing the rest of the sample. So this is, again, akin to seeing the needle without seeing any of the hay. Now, the range of light that these fluorescent tags emit is referred as the emission spectrum, and it's specific for each tag. And a filter matched to the tag's color is used to ensure only the light from the tag is imaged. So in this case, a green filter for a green fluorescent tag. However, biological processes involve multiple different entities, creating the need to visualize more than one target within the same sample. So a second fluorescent tag of a different color can be targeted to a secondary structure, and a filter matched to this color can be used. So in this case, a red filter for a red fluorescent tag. Now, each of the tags are imaged sequentially, and this enables us to build up an image. Now, the more color tags that we use of different colors, the more information that we can see from the sample. So consider here, we now use a third tag. So this is a yellow one, as well as the red and the green within the same samples. However, when you have multiple different fluorescent tags, the colors start to overlap and the filters are no longer able to work. So here you can see this overlap of the red tag into the yellow filter. So when we image the yellow tag, we now see this bleed through of signal. And this leads to this fake news as a issue in fluorescence microscopy. So essentially, the image from the red tag has now bled through into the yellow filter. And this is a contamination of the data. Now, this is a fundamental issue of fluorescence microscopy as it limits the number of biological targets that we can see. And this results in limited information from our biological experiments. Secondly, the hallmark of life is that it is animate. And to really understand biological processes, you want to see it as it happens, which is in motion. And this has led to video rate fluorescence microscopy employing cameras. Now, although this is a significant advance for the field, the speed at which you can image reduces as you image more fluorescent tags, as the colors are imaged sequentially for each time point. And this means that there is a delay between time points because we have to image each color independently, which leads to these jittery videos. So this means that biological information and events are missed. So ideally, we want to see subcellular phenomena in real time. So just to reiterate then, the two main issues that I'm trying to address is the fact that current fluorescence microscopy suffers from spectral bleed-through, meaning that we can only see 2 or 3 biological structures in our experiments, and also the fact that it is slow when imaging more than one structure within our sample. So, okay, to advance the field, I wanted to create a technology that fulfills the following criteria. Firstly, it should enable the visualization of many different biological targets to give more information from our experiments. Secondly, it should be fast, i.e., it should not image colors sequentially so that we can truly see what is happening and not miss anything. And finally, it should be a universal solution that can benefit any camera-based fluorescence microscope in the world. And this is exactly what I was able to achieve during my PhD. So this is a combination of hardware that I designed and built, as well as software that I wrote. And I'll now take the opportunity to very briefly introduce this technology and explain how it works. So consider we have a cell that's labeled with 8 fluorescent tags to different structures within its contents. So these tags are of different colors, and if we look at the spectra, they are highly overlapping, so we would never be able to image them using current technology. But what we can do is basically, we can hit all of these fluorescent tags at once using different colored lasers, and then we can collect the emitted signal and pass them through a single dichroic mirror. Now, these dichroic mirrors are very special in the fact that they can reflect colors that are bluer. So in this case here, you can see that the bluer signals are reflected and the redder signals are transmitted through. But then what we can do is pass these two spectrally separated channels through another pair of dichroic mirrors to gain the separation of 4 different colors. And if we do this for 7 dichroic mirrors, we can basically split the light into 8 different colored paths, and we can image them all simultaneously on 8 cameras. So in a nutshell, each of these 8 cameras image the exact same sample, just in different colors, and importantly, all at once. So it's much, much faster than current technology. We then take the data from these different cameras and we pass it through a novel spectral and mixing algorithm that I developed. And this reveals the true signals of the different biological structures without any contamination. Now, I don't have time to go into how the actual algorithm works, but this movie will demonstrate it. So essentially at the beginning, it starts off with an overlay of the data from these 8 different cameras, which is essentially a massive mess. So if I press play here, you'll be able to see the algorithm iterate and progressing as it works out what the underlying biological structures in the cell are. So by the end, we can truly reveal what the different structures are. Now, this is what the actual technology looks like. So this is the finished product that I designed and built, and importantly, I made it so that it's compatible with any camera-based fluorescence microscope in the world for maximum impact and improvement to the field. And here it is bolted onto 2 types of different microscope here at the MRC Laboratory of Molecular Biology in Cambridge. And I just wanted to share with you some of the first data that I was able to get using this system. So this is the first time ever 7 biological targets can be imaged simultaneously, in this case, in these cancer cells. And these movies are in the orders of multiple volumes per second speed, which makes our technology one of the fastest, most informative systems in the world for studying live cell dynamics. And this provides a huge amount of biological information. Now, I know this slide looks busy, so I'll just take a moment to explain what you're looking at. So essentially, on each axis is the biological structure name, and each panel represents the relationship between 2 of those structures at a time. So in a single experiment, you get all of this information, whereas using conventional 2-color time lapses, you'd have to do 28 experiments to get the same amount of information as imaging 8 compartments simultaneously using our technology. And each experiment would be slower because you would acquire the data sequentially. So ultimately, my technology saves the user time and money and gets them more information of better quality. Also, because all of this data derives from a single experiment, the user is less likely to suffer experimental variations. So this is a more robust, reproducible technique. Okay, now to show you some potential industrial applications, here are 2 collaborative projects that I've used this system for. So on the left, here you're looking at a movie of a mouse brain, and we were able to identify specific neuronal populations in different regions by visualizing specific gene expression, as seen in the different colors here. So we can use this for then looking at neurodegenerative diseases within the actual brain tissue itself. Now, if I press play here on the right, we can now see how different cell structures within cancer cells respond to various drugs. So in this case, the drugs makes these structures traffic and move towards a specific point within the cell. So although I personally will be using the technology for academic research, it could also benefit disease diagnostics, drug testing, and other applications. So the technology that I've developed has a number of benefits with no real drawbacks. Firstly, it is up to 8 times faster than conventional systems. Secondly, we can gain at least 4 times more information at no cost to the acquisition speed. And finally, this is an easy to implement system that can improve any camera-based fluorescence microscope in the world in a very simple plug and play fashion. Now, we currently have a patent application which is soon to be published for the technology, and we are finalizing a manuscript to present the initial results. And with that, I would like to thank my supervisors, Emmanuel Derivery and James Manton, for being massively supporting and guiding me through this project, as well as my lab members, various people at the MRC Laboratory of Molecular Biology, my wife, Savannah Kamari, for helping me design some of the slides, and most importantly, you for taking the time to listen.